moving on. We've got to describe, using diagrams and photographs, the histology and gross structure of the liver. Let's do with gross structure first of all. If we look down here, I've got a picture first of all of what the liver actually looks like. Okay, it sits in the upper abdomen just below the diaphragm, and it's supplied with blood from the general circulation in the hepatic artery. So the aorta feeds blood into the liver here in the hepatic artery. It also, the blood that's been processed by the liver then returns to the circulatory system. This would feed into the inferior vena cava, but also it has a blood supply that comes directly from the gut. Okay, and that it happens in this structure here, or this blood vessel here, which is called the hepatic portal vein. And this brings all the substances which have been digested and absorbed in the small intestine and the large intestine, and will bring it directly to the liver. Also to consider is bile. A substance called bile is produced by the liver, and we'll discuss later how that's produced. But it's stored in a thing called the gallbladder, and it is then released into the small intestine by the bile duct. If I were to represent that in a simple little diagram, just rearrange some of these words. Okay, so I've got the hepatic artery there, providing oxygenated blood from the aorta. I've got the hepatic vein taking blood back to the general circulation, the vena cava. I've got the hepatic portal vein here, which connects the gut to the liver, and I've got the bile duct, which carries bile from the gallbladder in the liver down to the gut. Okay, so that's a simplified diagram of what the actual liver looks like. Okay, so that deals with the gross structure of the liver. If we now go back to this and look at the histology, we've got to know about the histology, how the cells are arranged, and how the actual liver goes about what it does. If I look at the histology, first of all, if I look at the histology first of all, if I look at the liver under a microscope, what I can see is it's broken down into what we call a functional unit, which we call a liver lobule. So this thing that I'm circling with a cursor at the moment is what we call a liver lobule. Okay, this is a cross section of one, and you can see that in the middle there's a place where there isn't cells, and that is a vein, which is a branch of the hepatic vein, and then radiating out from the central hepatic vein, we can see that there are lots and lots of channels, okay, gaps between cells, which radiate out from the centre. Around the outside, we have collections of other vessels, two blood vessels, branches of the hepatic artery and the hepatic portal vein, as well as branches of the bile duct, which feeds bile into the gallbladder, and we'll discuss what bile is later on. Okay, so that's what a liver lobule looks like. If you're looking at these under a microscope, they're far easier to see in pig livers, because pig livers have a big capsule of connective tissue which separates each of these liver lobules. Not so easy to see in calf or human livers. If I now look at this diagrammatically, i.e. if I take a cake slice type sample of this to show you what it actually looks like in terms of a diagram of how the cells are arranged, I get this type of arrangement here, where we have in the middle, which is equated to this here, we have a branch of the hepatic vein. On the outside we have branches of structures. We've got the hepatic artery here, we've got the hepatic portal vein here, and then we've got a branch of the bile duct here. What happens is that blood arrives in the liver from the hepatic portal vein and the hepatic artery, and then it is funneled into these channels that exist between liver cells. Okay, so these structures here are all liver cells. We call these hepatocytes, and blood from the hepatic artery and the hepatic portal vein is channeled into these conduits or channels, okay, and pass along these channels towards the hepatic vein. These structures, the key word you must know, are called sinusoids. Okay, so the channels that link the hepatic artery, the hepatic portal vein, to the hepatic vein are called sinusoids. Key word, must have that in your notes. As the blood passes along between rows of hepatocytes, there's an exchange between the blood and the hepatocytes. So substances which are highly concentrated will diffuse along concentration gradients into the hepatocytes. Okay? Let's deal with bile. One of the substances which are produced by the hepatocytes is a substance called bile, which is a mixture of things called bile salts, and I'll talk briefly about what those actually are a bit later on. But as the hepatocytes produce the bile passes through the hepatocyte into these other channels, okay, and these are called bile canaliculi. Again, a key word. This is singular bile canaliculus, plural bile canaliculi, and the bile runs down the bile canaliculus towards the bile duct. Okay. Also, lining the sinusoids are these cells here. Okay, and they're called Kupffer cells. Another key word, Kupffer cells. And what they do is that they're phagocytic cells, and they engulf worn-out red blood cells, and they break down the red blood cells into amino acids, which can be used for amino acid or protein metabolism in the hepatocytes. Iron, okay, which is recycled and is taken back in the bloodstream to the bone marrow to make new red blood cells. But the heme group, the prosthetic group, which is called the heme group, the rest of it apart from the iron, is broken down into a substance called bilirubin, okay, which passes into the bile canaliculi, which then passes along the bile canaliculi, along here, into the bile duct. Okay, so imagine this Kupffer cell here encounters red blood cell, engulfs it phagocytically, breaks it down into amino acids, iron, which it can recycle and use, but the heme group is broken down and made into a substance called bilirubin, which then passes through these cells into the bile canaliculi, which then passes along to the bile duct. All the bile ducts converge into the gallbladder, and then the bile can be released into the small intestine. So, just putting some of those words together just to summarise all of that. If I look at here, so Kupffer cells. Kupffer cells, which are phagocytes, line sinusoids, engulf damaged red blood cells. The Kupffer cells then break down hemoglobin into globin, which is essentially the protein part of the molecule, iron, and the prosthetic group, which is called heme. The globin and iron are recycled, whilst heme is converted to bilirubin, which passes from the sinusoid, where the red blood cell is broken down, into adjacent bile cannuliculi. And then the bilirubin is released, along with bile, into the small intestine. And you break down millions of these red blood cells every day, so the activity of the Kupffer cells is very important. So just going back to the original slide here, which you have a copy of. That's in terms of the growth structure. Okay, so the key words that you need to realise what they actually mean. Hepatic artery takes oxygenated blood from the general circulation to the liver. Hepatic portal vein, this links the intestines with the liver and brings all the absorbed food materials from the intestines directly to the liver. Hepatic vein, which returns processed blood back into the general circulation, and you should be aware of the bile duct and gallbladder, which are involved with bile and transferring bile to the small intestine. Moving on to the histology, the functional unit of livers are these things called lobules. Okay, and as it said here, you should practice drawing one and labelling one, so you know what all the key words are. Okay, and the key words are all the ones which are highlighted here. Okay, so the centre of the lobule is a branch of the hepatic vein. Let's quickly flash back to that so we can show you. I've lost it now. So here we are, hepatic vein here, so hepatic vein, and on the periphery we have hepatic portal vein and the hepatic artery and the bile duct. Okay, other key words: sinusoid, it's a channel between rows of hepatocytes, and bile cannuliculi, which is also a channel which is delineated by two rows of hepatocytes that feeds bile towards the bile duct. Other key words: Kupffer cells here, these structures here, these phagocytic cells which engulf damaged red blood cells. Moving on to the next slide. So what you've got to do is describe the formation of urea in the liver, which we've already mentioned, including outline of an ornithine cycle, which is obviously something new to us. So as I said before, we can't store amino acids in the liver. Okay, what we do is remove the amino group. Okay, that's a process called deamination. Okay, key word as I mentioned before. Now the resulting ammonia is then converted to urea in a biochemical cycle, a bit like other biochemical cycles we've come across, such as the Krebs cycle, the Calvin cycle, and so on. It's a cycle of chemical reactions which are involved in producing things. Okay, in this case, it's a cycle which is an input of ammonia and carbon dioxide into the cycle, and an output of urea. Okay, and we'll just go through that in a little bit more detail. Okay, so as I said, ammonia essentially reacts with carbon dioxide. Okay, and it's a complicated cycle, and I'm not sure whether you actually really have to know this, although this diagram I've lifted straight out of the A-level textbook, so I suspect there is an expectation for you to know this. You should certainly be able to understand it. Okay, as I've got here, I've got a rather awful mnemonic, okay, which helps me remember the order in which everything comes, and I'll explain that in a second. So, as we said, ammonia is produced by the process of deamination, and what happens is that ammonia is fed into this cycle it reacts with a substance called ornithine, okay, and ammonia and carbon dioxide both feed in to this cycle. So the carbon dioxide feeds in, and the ammonia feeds in, and they react all together to form a molecule called citrulline. Okay, and citrulline is then converted to a substance called arginine, but another molecule of ammonia is also added to convert the citrulline to ammonia. Okay, and the arginine is then converted back to the starting substance, ornithine. But in doing so, we end up producing a molecule of urea. Okay, and that's called the ornithine cycle. Okay, I have a silly mnemonic: old and crusty Charlie, awful ammonia equals urea. So that's how I remember it. It's a rather silly mnemonic, but it works for me. Moving on. Now what we need to look at is the various roles of these hepatocytes. Okay, so we've looked at the structure of the liver, we've found out how the hepatocytes are arranged in liver lobules and how they are able to absorb materials which are in the blood. And now what we want to actually find out is some of what actually these liver cells actually do. We've already looked at how it deals with ammonia from deamination, converts it into urea. But now we need to look at one or two other examples. First of all, liver cells are rich in various enzymes, and some of these enzymes are involved in converting toxic substances into less toxic ones. The example you've just seen of ammonia being converted to urea is an example of that. But it's not the only example. You've probably often used the enzyme catalase, which you've probably found in liver, to convert hydrogen peroxide into oxygen, which is an enzyme assay we often use for working out the effect of temperature and substrate concentrations, etc., on enzyme activity. Okay, you might ask, why on earth is hydrogen peroxide involved in cells? And the answer is it is produced in cells during fat metabolism. So a lot of hydrogen peroxide is produced in cells, and as you know, it's a strong oxidizing agent. It's the substance that you use to bleach your hair, hence the term peroxide blonde. So it's obviously quite a poisonous substance, and it has to be got rid of incredibly quickly in cells, otherwise it would do considerable damage to cells. So there are enzymes in liver cells which convert hydrogen peroxide very quickly into water and oxygen. So that happens in hepatocytes. So just summarising that. So hydrogen peroxide is a metabolic waste, and so hydrogen peroxide broken down to oxygen and water by the enzyme catalase. There's an example there. Moving to other examples. Ethanol is a poisonous substance, and as we know, and is broken down in the liver by an enzyme in hepatocytes called alcohol dehydrogenase. And what that does is it oxidizes the alcohol and uses some of the molecule, or breaks it into acetyl groups, and feeds those acetyl groups into the Krebs cycle, involved in the link reaction into the Krebs cycle. Those acetyl groups are fed in for respiration. So some of the molecule yields ATP. Okay. And I've also written here, too much alcohol reduces oxidation of fatty acids in the liver, which then deposit in the liver to produce fatty liver. These acetyl groups, which are produced by the breakdown of alcohol, are also produced by the breakdown of fatty acids, and these acetyl groups are fed in to the Krebs cycle. So if you immerse your alcohol dehydrogenase enzymes with alcohol all the time, what happens is that you don't break down fatty acids, and therefore that fat is stored in the liver, and that's called fatty liver disease. It's also, if you're on a diet, one of the things they say you should never do is drink alcohol, and that makes sense, because if you're drinking alcohol, you are producing these acetyl groups which are being fed into the Krebs cycle, which means that you're not breaking down fatty acids. And obviously you're not breaking down fatty acids, you're not using fat, and therefore you're not going to lose any weight. Okay, so that gives you a sort of biochemical reason why you shouldn't drink alcohol if you're trying to lose weight. So, in summary, the ethanol broken down in liver cells by an enzyme called alcohol dehydrogenase. Acetyl groups are fed into Krebs cycle via acetyl coenzyme A, which you'll come across when you study respiration.